All right, so today we're going to talk about genome assembly. And okay, so what we're going to talk about today is essentially what we're looking at on the right. The picture there is from a paper by von Iverson, who did one of the first metagenome assembled genomes from a eukaryote in the Lake Washington. And each of the colors here in this assembly graph corresponds with a certain taxa. So he used this to pull out one of the first genomes from an unsequenced organism, from an uncultured organism from the environment. Using some software called C-Star that I don't think anyone uses anymore, but it was a first of its kind. So we're going to talk about how to do that today. First, we're going to talk about sequencing statistics and how to calculate how much sequencing you might need for your samples. If you're working in acid mine drainage, it's a very different story than if you're working in some complex sludge or soils or something. And then we're going to talk about different paradigms for genome assembly. A primary one called overlap layout consensus, or OLC, and another one called the Bruijn graph assembly are the most common ones used today for metagenomes, but there's other assemblers used for other kinds of data, for example, long read data, and you can combine multiple of these in methods called hybrid assembly. So we're going to touch on those just a little bit at the end as we talk about assembly software that's out there. There's no shortage of software out there to do this stuff. So just as a reminder of what we're working towards here, essentially, for the most part, we get data in the form of short reads of chopped up genomes. And our goal a lot of times is to reassemble those into complete genomes, or as near complete as we can get. So a question we might ask is, do we have enough coverage here to piece together the red genome? Are there enough red pieces there that we can link them all together and form a complete genome, and then do some functional annotation on that and find out what that genome is up to in the environment? So the answer to that question depends on the relative abundance of that red genome in the environment. So if we have this collection of cells over here on the left that makes up some population, and we extract the DNA and do a library prep, which involves randomly shearing the DNA into small fragments, then we get this genomic library over on the right. And we'll see in this library a reflection of what was in the original population. So species like the red taxa, which are very abundant, will have more coverage in the library compared to moderately abundant species or rare species, assuming that their genome sizes are all about equal. If you have some diatoms in there, say you have an equal number of diatoms in bacteria, then obviously the diatom, which has a genome size maybe 100 times bigger than the bacteria, will outweigh that. So when we're just looking at prokaryotes, it's a pretty good estimate that what you get in your library will reflect what is in the environment, but that's not always true. Another thing that can affect that is your DNA extraction. There have been a lot of studies looking at different DNA extraction protocols and how that affects what you end up with in your genomic library. For example, if you have a lot of spores in your population, they may not be extracted well, and so your genomic library could reflect the absence of those. The same with fungi are often not extracted well by typical DNA extraction protocols. So if you're particularly interested in some taxa, you have to make sure that they are well extracted by your DNA extraction protocol. And the final thing I guess I would say is that what you can do with the data afterwards will depend on the coverage you get. So if you're looking at the abundant groups like this red one here, you can probably get a pretty nice draft genome assembly. Whereas if you're looking at the rare groups, maybe the yellow ones, it may only be possible to do some kind of read-based analysis, either mapping or direct annotation of the reads. So you can see that when you're looking at the evenness as well. Community diversity is often described as both a function of richness, which is just the raw number of taxa that are present, and on the evenness, which is how the relative abundances of the taxa vary inside the community. So on the sample 1 at the top here, we have a very uneven community where this first blue taxa has much higher relative abundance compared to the other taxa. And so when you look at the DNA pool on the right, you have a much bigger slice of blue pie than yellow pie, say. In the sample to the left, even though the overall relative abundances are the same in our DNA pool, I should say, the amount of DNA in our DNA pool is the same. The distribution is very different because the evenness of the sample is much different. It's much more even. And so the first blue taxa has about the same relative abundance as this dark blue taxa. And so what we get in the DNA is the same. So this is a reminder, again, that what we get out of the metagenomic sequencing is compositional and not raw, unless we're doing some kind of special spike in so that we can do a back calculation later. But normally what we get is compositional data or proportions. So different environments have different amounts or gradients of diversity, and different environments will require different approaches for answering the questions that we have about what the microbes are doing in those environments. So low diversity environments like cultures or symbiotic associations, those are very much more amenable to assembly-based analysis. Also, extreme environments like acid mine drainage can be very low diversity environments, and so relatively easy to assemble from. As opposed to environments like up here on the right, where we have either raw seawater or soil or activated sludge. These kind of samples are very difficult to get complete genomes out of, especially if you have a relatively high evenness in the community. And so those tend to be more amenable to things like read-based analysis, which doesn't require the complete genome to get interesting answers out of. So a lot of real environments that we're interested in are the more highly complex environments. So it might be like this, where you have hundreds or even thousands of taxa in here. And you may know this from previous experiments that you've done using 16S sequencing or fish, for example. You may know what this should look like before you even do the metagenomic sequencing experiment. And if you do, and you're interested in one of these very rare taxa, like this little red sliver here, it may be worthwhile for you to do some kind of preliminary exercise of enriching that microorganism. If you know that it grows on some particular substance, you could throw that in and do a little mesocosm incubation and try and increase its relative abundance. You could do things like cell sorting, where you label it with a fish probe and then sort out those cells into a particular bin, or sorry, into a particular well of a sequencing plate, and then sequence that enriched community. Or do single cell sequencing, where you can sort individual cells into individual wells of a plate and then sequence them individually. So there are methods for getting at these rare taxa. We're not going to talk too much more about them here, but just know that if that's something that you need to ask in your scientific experiment, then there are ways to address that. Okay, so let's say we have kind of a typical community, and we are interested in questions like, how much coverage do I need to get a complete genome out of this environment? So the first thing we need to know is what actually do we mean by coverage? And so there's an equation here, coverage C equals L times N divided by G, where L is the read length, N is the number of reads, and G is the haploid genome length. So that's the the number of base pairs in one copy of the chromosome. So as an example, if we're going to sequence E. coli, which is a genome that is about 4 megabases, 4 million base pairs, and we're going to do it on a whole MiSeq run using 2 by 250 base pair chemistry, then we have a C equals L, which is the read length of 250 base pairs, times the number of reads. There's 8, you can get about 8 million paired end reads from MiSeq run, divided by the length of the genome in base pairs, is about 500x coverage on average for this genome sequencing project. So there are some questions we can ask about, is that enough? And how do we know if that's enough to actually get the complete genome that we're looking for? This does make a couple of assumptions. One is that the genomes are randomly sheared during the DNA extraction and library prep, and that is not always true. Sometimes there are biases introduced at that step. Some of the library prep protocols include the use of a transposon, which is biased towards or away certain parts of the genome, for example, GC-rich genomes, parts of the genome. And so you can introduce bias at these steps where they might actually influence the assembly that you get out of this. There's also an assumption that during sequencing, a random subset of the fragments are sampled, and this is typically true. So we have more questions we could ask. So what is the probability that a base will be sequenced exactly 0 times given some coverage? That's essentially asking, what's the likelihood that we will be unable to complete this genome sequence because there will be at least one base that remains unsequenced? And so the way we answer this is using the Poisson distribution, which is a statistical distribution that explains what happens when you are randomly sampling from discrete cases. So on the bottom right here, we have a graph of the Poisson distribution at various functions of K and lambda here, where K is the number of times that a base is seen in this case, and lambda is the average coverage. So up here we ask the question, what's the probability that a base will be sequenced exactly 0 times given a coverage of 4? So the coverage is lambda here. We can look at lambda equals 4 and just read off the graph here what we would expect those probabilities to be. So we can see here that K equals 0 and lambda equals 4. We have a dot that's somewhere above 0 and below 0.5. The average you can see of the distribution is here at 4, where we saw our average coverage is that we're asking about. So what this means is that there are some fraction of bases that are sequenced 0 times, some that are sequenced once, twice, 3, 4, 5, 6, 7, 8, some even 20 times at some very low rate, just because of this random sampling that happens during the sequencing process. So there's an exact equation over here on the left that you can use to calculate this. The probability at x equals k equals lambda to the k, e, where e is Euler's number, to the negative lambda over factorial k. So if we want to plug in these numbers down on the bottom right, the probability that a base will be sequenced exactly 0 times at a coverage of 4 is the probability at x equals 0, which is 4 to the 0 times e to the minus 4 over 0 factorial. 0 factorial is defined as 1. 4 to the 0 is 1. So this comes down to e to the minus 4, which is 0.0183 or 1.83%. So there's about a 2% chance that at a coverage of 4x of your genome, you would have about 2% of the bases would be unsequenced. Obviously, that's not ideal, so we should probably want more coverage. And sometimes we're looking for a minimum coverage or some kind of coverage goal. So if we wanted to say, for example, what is the probability that a base will be sequenced 2 times or less, given a coverage of 4, we could just add up the probability at x equals 0, x equals 1, and x equals 2, and find out that there's about a 24% chance that any given base would be sequenced less than 3 times. So we will want to do better than this when we're actually doing the genome sequencing if we want to get a good assembly. So we can ask it another way: what is the minimum coverage necessary to ensure statistically that no bases are left unsequenced? For that, we can do something like set the probability to a very low number, for example, 1 in 4 million. That would assume there was only one base that was unsequenced in the entire E. coli genome, and then solve for k, or solve for lambda, the coverage. So in this case, we have the probability 1 over 4 million equals coverage to the 0, where the 0 is the number of times it's the base is seen, equals 1 over 4 million, sorry, 1 over 4 million then equals e to the minus c. So we can solve that by taking the log of both sides, and we get that c is about 15.2. So to sequence this E. coli genome and assume that only one base remains unsequenced, we need a coverage of 15. So if we have a coverage higher than that, we assume that all of the bases are sequenced. So when we're thinking about metagenomes now, we have to multiply this by the number of cells that are in there and the evenness, and you could do some more calculations there to find out what the read depth would need to be if you wanted a complete genome of some organism that you have reason to believe is present, say, at 10% of the community. So you can actually get those numbers. So how do we actually look at coverage in metagenomes? This has been a question that people have asked for a long time, trying to figure out, okay, if I keep sequencing this sample, will I always find new k-mers that I haven't seen before? So this is an example from a blog post in 2012, just showing the number of sequences on the x-axis here on the left, and the number of distinct k-mers in millions at a length of k equals 31 for a variety of different samples here. The bottom one is E. coli. So about 10 million reads will essentially saturate your k-mer curve for E. coli. And you could ask, why isn't that number closer to 4 million? Because that's the number of k-mers that should be in a genome sequence that's about 4 million base pairs long. And one of the answers to that is sequencing error. So every time you get a new sequencing error in your sequences, that essentially adds 31 k-mers to your data set. So you can also use this almost as an estimate of the error rate of your genome sequencing. And that'll come in important later when we're talking about assembly. So they have some other examples here. So here's in red some compost from a compost facility. The green is a lake metagenome, and the blue is a field, like a pasture metagenome. And you can see that as you get to these more complex environments, there's no sign whatsoever that these are going to saturate anytime soon. It looks like the more you sequence, the more k-mers you find. And so people have since then done ever deeper sequencing. And the paper on the right is from 2018, discussing a software program called NonPareil, which looked at statistical analyses of metagenome coverage at different efforts, so at different sequencing amounts, and tried to predict from subsamples of that how much the total diversity in terms of k-mers was in these samples. So if you look at these graphs, particularly this one on the bottom right, which shows the k-mer length on the x-axis, and then the projected effort needed to reach 95% coverage, and that is to sequence 95% of all the k-mers that are actually there for a variety of different samples here. So there's some body parts here, there's some stool, there's some natural lake samples, there's a soil sample here. So the soil, for example, it looks like it would take about a terabase of sequences to get 95% coverage of this sample, compared to a lake, which may only take 20 or 30 gigabase pairs. A entire NovaSeq run can be something like a terabase or two, and that's $20,000. So if you have 20 grand to spend sequencing a sample, then you can completely sequence the k-mers in this soil sample, but a lot of times you don't. So a lot of times we do multiplexing, where we do barcoding to run a bunch of samples on one run. For example, you can run 384 samples at once, in which case your overall coverage will be less. And so this is just a matter of finding out what your questions are and how much depth you need in your sample to do what you want. If you're interested in these very rare taxa, then you may need a very high sequencing depth in order to get at your taxa of interest. If you're just interested in star 11 in the ocean, you don't have to sequence that much because it's 33% of the cells wherever you go. So there's different sequencing depths required depending on what your scientific question is. So don't always just imagine that more is better. That's not always true. You can always think about it a little bit and use your resources more efficiently. So that was just looking at k-mers, and we also talked about how k-mers can be introduced through sequencing error. So one way to look at these without using k-mers is to generalize a little bit and look at something like genes here on the left, or taxa on the right. So the graph on the left is looking at the number of non-redundant genes, so using a gene caller and a clusterer to assign genes to gene families, based on the number of samples sequenced for various different sample types. So this lowest one here is from mice, and the red one is from, I actually forget what these abbreviations are, but the yellow one here is humans. So you can see after you sequence 1,000 samples of whatever this is from humans, you see a leveling off of the number of unique genes that you get. Here, there's something like 10 million. But you have a faster growth rate at the beginning. So if you wanted to sequence all the genes that were in this environment, you may need to sequence either some samples very deeply, or just a lot of different samples and get the coverage that way. On the right, we're looking at the percentage of shared taxa in this case, based on different taxonomic levels, from genus to phylum, and looking at the number of individual samples. So this is another way to look at it that's more like looking at the core of these. So what's present in every single thing that you sample. So you can see that the same phyla in this purple line at the top, as you sequence more samples, there are fewer that are present in every single sample. Whereas if you're looking at genes, there's a rapid decline as you sequence more samples. So there's only a few genes that are present in every single one of these samples. And that's another way to look at how much more sequencing you need to do if you're really going to characterize an environment. You just have to know that sequencing 1 or 2 samples from any given complex environment is not necessarily representative of that entire environment. And it may be that you need to sequence a whole bunch of samples in order to get good statistical estimates of what's actually in those environments. And that's something that people doing metagenomics have not always done, and it's something we're learning how to still deal with, because a lot of times we are resource limited. So we say, oh, wow, we got 10 metagenomes from this environment. That's pretty good. But there's probably a lot more that we're leaving behind that we're missing. So one of the next phases in metagenomics is massive scale metagenomics, where we do sequence hundreds or thousands of samples from some environment and look at these variations, then gradients in diversity and distribution over time and space and stuff like that. So I'm going to talk a little bit about the software that's used for assembly. And like I said earlier, there's no shortage of it. I would also say that I'm not an expert on assembly software. I tend to go with a couple of packages that I know work well for my environment. It may be that for your data set and your environment, you will need to look into papers of people who've been doing this recently and see why they chose specific software packages, specific preparation pipelines in terms of quality control or error correction, specific flags used for each of these. A lot of them have an isolate version and a metagenome version optimized for either relatively even coverage of genomes, which would be for the isolate case, or very different coverage in the metagenome case. And it's also a very rapidly evolving field. So if you're looking at a paper from 5 years ago, it may not be that relevant. You should probably try and find something much more recent in order to find out what the latest, greatest is, because that really will affect your results, both in terms of their quality and probably in terms of the computational resources necessary to do the assembly, because the algorithms are getting faster and more efficient over time too. So there's kind of three classes that we've broken them down here to. On the left is the OLC assemblers, which were originally designed for like the Human Genome Project, and which used Sanger sequencing, which had very long, high quality reads. And my understanding today is that for most of these, they're used still in eukaryotic genome sequencing. I haven't used any of these ones on the left, which is why I say that. But if you know different, feel free to speak up. And we'll go through in brief detail how these different assemblers work after this. In the middle are the Bruijn graph assemblers, which are optimized for the high quality short reads that are generated mostly these days by Illumina sequencing. And they're the bread and butter for prokaryotic metagenome, or for prokaryotic isolates and for metagenomes. And in particular, I just wanted to mention the three that I've seen personally most used in the world of metagenomics, especially large scale metagenomics, are SPADES, MegaHit, and IDBA. And each of these has a version that's specifically tailored for metagenomes. So for SPADES, it's called MetaSPADES. For MegaHit, there's a flag for running it as a metagenome. And for IDBA, there's a fork called IDBA/UD, I think. But some of these other ones have been used also recently in a project called someone want to help me out? The Metagenome Comparison Project. CAMI, thank you. Yeah, I have a slide on it later, but I spaced out. So there's a lot of interest in the community about how to evaluate the quality of these assemblies, since a lot of times the environments are too complex to have any kind of gold standard. So I don't know, maybe for acid mine drainage, there's a gold standard that is used for knowing exactly what organisms are your environment and what their genome should look like. But for most environments, there's nothing at all like that. So when you don't have a gold standard, it becomes tricky to compare the quality or the usefulness of different assemblers and assembly pipelines, because there's no best thing that you're aiming for, that you know. And so there's a project called CAMI where people are trying to use reproducible pipelines in order to try and provide some valid criterion to evaluate both the assemblies themselves and also the software that's used for the assemblies. So if you're interested in finding out more about that, you can look up CAMI, C-A-M-I. Finally, on the far right, are the long read assemblers, which are optimized for low quality long reads from PacBio or nanopore sequencing. So nanopore is getting better, but the qualities are orders of magnitude worse than Illumina. I think Illumina sequencing has error rates of something like 1 in 100,000 or 1 in a million. And nanopore sequencing often has errors on the order of 1 in 10 or 1 in 100 or 1 in 1,000 if you have a lot of overlap. And nanopore also has particular problems with things like homopolymers. I don't know the error profiles for PacBio, but these software packages on the right are optimized for these error profiles and for these using long reads in your assemblies. And we'll talk a little bit more later about how that works. But if you're doing nanopore, you'll want to use those. So here's a paper that did a comparison of some of these assemblers using real metagenomes. So on the left is a kelp biofilm, on the right is a forest soil. This is from Vollmer's paper, et al. 2017. And in this figure, they're just counting up the number of marker genes they found in their assembly. So that's a useful metric for just looking at how much of the functional gene content made it into the metagenome that was available. And so you can see again that these 3 that tend to stick out are IDBA, megahit, and metaspades. So that's another confirmation for me that I don't need to really investigate these other ones because they also work in my samples. But again, your samples might be different and there may be reasons you might want to investigate these other packages. Okay, so I'm not going to go into the assembly theory in great detail. At the end, I will provide some links to YouTube videos by Rob Edwards, who's an expert in this and who teaches a class on it. And so if you want to go more into the nitty-gritty about the theory for sequencing assembly, there's a lot of really interesting stuff in there, but I'm not going to go into extensive detail in it. I'm just going to basically show some of the differences between the different algorithms and what that might mean for you. So in this example, we have the 2 major types here. So on the right is the OLC, the overlap graph method, and then on the bottom is the de Bruijn graph method. And they're both ways to link together subsets of the data in such a way that you can put them into some kind of proper order. So on the left is the read layout as given in if you were to do an alignment. And you can see that we have reads, they're 2, 4, 6, 8 base pairs long in this case, and they cover some fragments of DNA here. These blue ones are R1, R2, R3, R4. The singleton S, which I added, and then A, B, and C are these green ones, and X, Y, and Z are the purple ones. Excuse me. So using the overlap method, essentially what you do is an all-by-all comparison where you look for regions of overlap between every read and every other read. In this case, we have a minimum overlap length of 5 base pairs. And so you can just do some kind of blast or other kind of like Smith-Waterman algorithm to find out all of the places of overlap between all of your reads. And you can imagine this gets very computationally intensive, having to do any kind of all-by-all comparison. But it's the way the human genome was originally assembled, which was a massive feat. And so it does work, but it just takes a lot of computational effort. Because essentially what you get is these nodes, which represent the reads here, and then all of the edges that connect every read to every other read that has some overlap that exceeds the minimum length. So in this graph here, we have the minimum overlap of 5. There's 10 nodes, 17 edges, and this one singleton, which has, as you can see, either an error or a SNP. It may depend on what's going on, but it has a G here instead of a C. And you can see it's 1, 2, 3, 4 bases here and 5 bases here. So it has no 5 bases in common with any of these other reads. And so it's unassembled on the overlap graph. It's just left as a singleton. Now, if we look at the De Bruijn graph on the bottom, it uses a different technique. So in this case, we go to our old friend, the k-mer. We break all of these reads up into their constituent k-mers. Here we're assuming a length of k equals 3. So in this case, we break up our reads into GAC, ACC, CCT, CTA, blah, blah, blah. And then we can do the same kind of graph assembly as before, but in this case, instead of looking at the overlaps, we're just looking at the k-mers themselves. So here we have GAC is connected to the k-mer ACC, which is connected to the k-mer CCT, etc. So if you wanted to read off the final sequence from this assembly, what you do is look at the first letter of each of these k-mers. So you can read this off as G, A, C, C, T, A, C, A. And then you get to a fork where you have these two different reads that both have this k-mer AGT, but then they separate off into different directions. So then we have GTT, TTA, TAG over here, and different bases down there. So this is called a fork, and it's very common that there is a stretch of shared DNA between two sequences that are in different parts of the genome or in a metagenome in different genomes all together. And so this is not uncommon that you would have a fork like this. What would happen after this in the genome assembly process is that these would get cut into contigs. So essentially at the end of this, we would have a blue contig from GAC to AGT here. And then there would be a cut here, and we would have a contig that was green and a contig that was purple in our output files. And so this is one of the issues that we come to when we're doing metagenomic sequencing and assembly, is that the assembly graphs become very complicated, and especially for close related sequences where there might be a SNP here in this case, that will tend to break up the contigs into much smaller pieces than you would have in the absence of those SNPs when you have closely related genomes. And it can break up the contigs so much that they may not even appear later in your binning process if you have a minimum cutoff length. So there's all these issues that come back to the assembly process in our downstream processes related to binning. So I was just going to finally mention that the singleton here, the orange read, is integrated into this De Bruijn graph as a bubble. So you can see there's kind of a ring-shaped structure here, and that's called a bubble in De Bruijn graph lingo. And so a major part of the assembly process is to go through and pop bubbles when they appear, if necessary. And one way the assembler does that is to decide if that bubble represents an error or a real feature in the genome. So the overlap layout consensus method takes individual sequencing reads, finds all of the overlaps, and then matches them together into the longest contiguous DNA fragment it can. But this assembly process is very much complicated by heterogeneity in the sequences. For example, if you have a diploid genome where you have different isoforms on each strand or on each chromosome, then that can make it very complex. It also makes it hard when there's undersampling, because gaps in coverage can really affect the final length of your contigs. The other thing is that unassembled reads that do not find singletons or do not find overlap can show up as singletons, and that can lead to the loss of some of the information that's present in your raw reads. For the de Bruijn graph assembly, what you'll see as we do the assembly later are some of these steps. The first thing that happens is that the k-mers are indexed into a hash table. So we have all of these reads that are then broken into k-mers, and the length is typically on the order of kind of 21 to 127. They're generally odd to avoid palindromes. And those are put into a big hash table in memory. The next thing is to step through those k-mers and collect them into nodes. So that's finding all of these all but one overlaps that we saw previously. So finding the GAC that has an overlap with the ACC as AC. So it'll take the, if we say 21 mers, and find all of the reads that have the last 20 bases, or sorry, all of the k-mers that have all of the last 20 bases in that exact order, and add them to the node and do kind of a single leakage clustering to find all the nodes. Then the assembler will look at the read coverage, so do some kind of mapping to find out the coverage of each node, because that can help you figure out which bubbles to pop and which bubbles to keep, things like that. Another task in the assembly process is often to rescue k-mers that would otherwise be lost. So in order to avoid this making of bubbles due to sequencing error, a lot of assembly pipelines just throw out singleton k-mers, or they don't include them in the primary step. And that's to avoid the unnecessary complexity of the assembly process. But it's the way they do that is often to throw out the entire read and any k-mers that are on that read. So what this process does in MegaHit, for example, is if there are some solid reads that do have good coverage, they will go back and include all of the k-mers that would have been thrown out by the coverage limit back into that. So you'll often see this in assembly processes. There are also mechanisms for flagging repeats, which have unusually high coverage, assembling prefix and suffix nodes to create the edges, simplifying the graph by collapsing the nodes into long linear stretches when they're available. And then the step that makes the assembly work is to find a path that visits each edge only once. So what you end up with is a very complex graph, and the idea is to essentially pull that apart in order to make the longest contiguous sequence that visits each edge in the graph only once, and then output those as contigs. So in very, very quick terms, that's how the de Bruijn graph assembly works, and people are constantly getting more and more clever at doing this in order to get the best possible assemblies out of your data. I wanted to talk just a little bit about long read assembly. For one, because it's really cool. It's helped me in my own research to get complete one contig circular genomes from bacteria. And I think it's a really exciting technology. I'm not going to go into the technology, but just mention that there's two ways typically that long reads are used in assemblies. One is in a hybrid approach, where you use the long reads as scaffolds to resolve repeats from the short read assembly. So with 15 or 20x coverage, you can usually get a quite good short read assembly. So if you do Illumina sequencing, let's say you can sequence 25 decent quality isolates on one MySeq run. That's pretty good. And then you can use long reads to scaffold those short reads contigs such that you can span any repeats that were unfixable by the Bruijn graph assembler. The second is to use just the long reads in a de novo process, which generally requires a high coverage to resolve the errors necessarily. But you can do a de novo assembly with just the long reads. And then often what people do is a polishing step where they take either good quality contigs that they know from previous assemblies or from references even, and turn those de novo assemblies into higher quality assemblies. So you'll see different long read assemblers will have preferences for one of these methods. Spades, for example, is one I use to do a hybrid assembly with the scaffold approach. So there's a lot of questions to be asked about how do I get the best assembly from my data, either a metagenome assembly or a isolate genome assembly. And sometimes it's by luck, and sometimes it's by looking at the literature and see when people have done the past. And sometimes it's just by trying a bunch of things and finding out what works the best. So figuring out what the most important parameters are that you might adjust in whatever software package you use, and then just running it a bunch of different times and using some quality control software, which we'll talk about in a couple lessons, to figure out which assembly works best for you. So all of these software packages have tons of options that you can change. For example, the length of the overlap or the Kamer length. There's oftentimes penalties for looking at mismatching gaps in the OLC assemblers. Quality scores. There's also all of the factors that go into inserting the reads that you use for the assembly. So Spades has a built-in error correction module called Bayes Hammer, which does error correction prior to the assembly. You can turn it off if you're using your own error corrected reads, but it tends to improve the assembly to do error correction. So it's often an important step before you get to the assembly part. Generally, the outputs from assembly programs include some kind of FASTA files with the contigs or scaffolds if you're using paired end reads. And some of the software outputs assembly graphs. And I'm personally a really big fan of assembly graphs. And I would urge you, especially if you're doing isolate genomics, to always look at your assembly graphs. This is one on the right of Polaribacter that we sequenced in my lab. And one of the nice things about looking at the assembly graphs is that you can very easily tell if your genome is complete. So this genome is not a single contig, but you can tell that it's complete. It doesn't have any loose ends, essentially. You can also tell what contigs did not make it into the genome, either because they are maybe a plasmid or because they are contaminants or untrimmed adapters or just some other kind of artifacts, and you can throw them out. So that's nice. I also look at my metagenome assemblies in the graphs, and I find it really helpful for pulling apart some of these genomes that I'm interested in. So one way to look at this is a metric that I call finishedness, which I just kind of made up. Probably has a real name somewhere, but it's the size of the largest connected component in the graph divided by the total size of the assembly. And I use this to help me find out what kind of coverage I should be aiming for in my isolate genome assemblies. So here's some examples where we have a complete genome in one connected component. In graph speak, that means that all of the contigs are connected to each other. So 100% here. And that genome had an average coverage of 120x. Some of these genomes are kind of in the middle. Most of the assembly is in one big connecting component, but we have these other pieces that are disconnected. And so they might fall over here somewhere. And then this is often what we have in the world of metagenomics, where we just have a whole bunch of contigs that are disconnected from each other. And so those would have a low finishedness on this metric. And that's often what we face when we're doing real life metagenomics. So earlier we estimated that we would need a coverage of about 15 to get a complete assembly of E. coli. In real practice, it often takes more than that because of various imperfections due to biased preparations in the library or in DNA content for various reasons, like exponential growth. If you have cells that are growing very quickly when you do the DNA extraction, you'll find that there's higher coverage around the origin of replication, where that active replication is happening, than on the opposite end of the chromosome. And people actually use this in to try to predict the growth rates of environmental organisms from metagenomes. And I'm not sure if it works or not. I think people are still trying to figure that out. So here's some examples of what you might expect from the Bruijn graph visualization. And I'll lead a tutorial on this using the program called Bandage, probably next week. So if you're interested in this, come check that out. But what this is showing is an assembly of two very closely related genomes. So these are essentially different strains of the same species that have an average nucleotide identity of about 97%. So what you can see is that every time there's a single nucleotide polymorphism between these two genomes, we get a bubble. So the bubbles are in black because they're lower coverage, and the pieces of the genome that are contiguous or identical between the two genomes are in red here. So essentially we have these red, black, red, black, red, black, red, black pattern. And each of these, the lengths of these contigs in this assembly are something like 100, 200, 300 base pairs long, because there's so many SNPs in this genome. And so what that means is, if I was using this for binning, where we use a mapping protocol, which generally has some kind of cutoff of about 1,500 or sometimes 2,000 base pairs as a minimum contig length, these two genomes would just disappear, because all of the contigs here are less than 1,500 base pairs. So this would, even if these were very highly abundant in the environment, and it's often the case that highly abundant organisms have a lot of interstrain variation, they would just not show up in the bins. And so you might either need to do something special to pull those out, or you might need to do some kind of read-based analysis instead of an assembly. Sometimes you can pull them directly out of the metagenome assembly, though, from the graph view. So that's what this is. This is some portraits from a 100 megabase pair metagenome assembly that I pulled out. It's from seawater. And you can see that these assembly graphs can get very complicated, especially. I think these are probably eukaryotes or some very closely related bacteria. For example, gamma proteobacteria tend to have a lot of overlap with each other. You also see big clusters of 16S ribosomal RNA genes in this kind of thing. This is basically a whole bacterial genome that's shown up as a single connected component in the assembly graph. So I find them as very useful both in isolate genomics and in metagenomic analyses, even though they take forever to load on my laptop. Okay, so I just wanted to point you to a couple more resources. Here's a good lecture by Will Trimble on looking at k-mers to find out how to assess the completion and the quality of your metagenomes. Here are the videos by Rob Edwards about the assembly process. Here's how MegaHit defines Mercy k-mers, in case you're interested in that process. And here's a link to the CAMI, the Critical Assessment of Metagenome Interpretation. And if you invent a great new either assembler or binner and you want to test it out against the best in the field, there's an open challenge to do that. So you can go to the camichallenge.org and submit your own data and pipeline, or submit your own pipeline to analyze their data sets, which is a pretty fun process. Okay, so after this, we're gonna jump into the tutorial. So if there's any questions from the assembly process, you can shoot them at me now.